This D2 molecule that you have, you show that in macrophages, when you activate them with interferon gamma and LPS, the channels appear. What happens if you treat those macrophages with the D4? What are the consequences to the macrophage? Because I was surprised that in your list of chronic inflammatory diseases, you didn't put arthritis, for example, which would be the obvious disease to think about.
Yes.
What's the consequence on an activated macrophage if you give the inhibitor that of the hemichannels?
Yeah, the macrophages, the release of cytokines requires hemichannel activation. If you block the hemichannels, macrophages will not release cytokines. So you reduce the role of macrophage in inflammatory response. But the important thing is all this inflammatory disease is the inflammasome that is present in the parenchymal cells. For instance, in denervate muscles, these denervate muscles show inflammation with no cytokine being released from macrophages because there is no infiltration of immune cells. So the main problem is the inflammasome, not the inflammatory cells. And the inflammasome is completely independent. Now, the other example that is very interesting to remember is that in clinic, dexamethasone, which is an anti-inflammatory drug that's frequently used, blocks macrophages, polymorphonuclear cells, and chronic treatment with dexamethasone is toxic and causes muscle atrophy. We just demonstrate that the atrophy is due to expression of hemichannels in the muscles. If you do the same experiment in animal knockout of connexin 43 and 45, there's no atrophy. So eventually, it will be needed to modify the anti-inflammatory treatment by giving dexamethasone plus hemichannel blockers, and that will be a more optimal treatment.
Yeah, well, that was what I was trying to get at. What are you actually achieving in terms of giving an inhibitor of the hemichannels? Because you go from the macrophage activation to very complex diseases.
Yes,
and
yes.
The dexamethasone treatment is obviously not a comparator for it, because we know what dexamethasone is doing and why it's toxic. What I'm trying to see is what is the specificity of the action that actually you achieve to have as a result an effect on a chronic inflammatory condition, but you don't mention arthritis, for example.
Right, the specificity is given by the cell itself because under acute condition, the hemichannels are not so critical, but under chronic condition, the hemichannels become expressed and very active. And as I showed, the hemichannels are necessary for physiological responses, but if you work the pharmacology, you can block the overexpression or the overfunction of this hemichannel without affecting the basic condition. So is that the specificity? Block hemichannels that appear under chronic conditions in parenchymal cells, not necessarily in the inflammatory cells.
So an atherosclerotic plaque, for example, is full of these hemichannels. That has been known for some time, and probably they vary in their expression at different stages of the plaque. What do you achieve? What happens to the plaque if you give one of these, your compound? That's the question.
We haven't studied that yet. This is many of the results that I show you in muscular dystrophy with an epilepsy with D4 is unpublished data. It's all patent, and we have a spin-off related to that, but we haven't published. We hope to publish this by the end of the year or next year. We were holding this data because of patent reasons.
The next question is by Gabriel Rabinovich.
Okay, very nice talk. So you talk about chronic inflammatory disorders, and one of the most important processes in chronic inflammatory disorders, particularly during neurodegeneration, is the polarization and conversion of microglia into M1 microglia, classically activated microglia. So my question is whether there is a function of hemichannels and intercellular communication in this classically activated microglia that produces high amounts of nitric oxide and low amounts of arginase, whether there is an interaction between astrocytes and microglia in polarization of this M1 microglia.
Right. We haven't studied, nobody that I know has studied the polarization response in this chronic inflammatory disease. But you brought a very interesting issue that I think is very interesting from the social point of view, especially in stressed mothers. Okay, the microglia remain active after stress during pregnancy or early after birth, and not the astrocyte or oligodendrocyte, but the microglia remain active and they release pro-inflammatory molecules. So the development of the brain is altered. And this may be the explanation of many psychiatric conditions like depression, schizophrenia, and so on, because the connectivity of the neuron doesn't occur in the right way and the migration doesn't occur in the right way. So a new stress condition will activate the system and the patient shows the symptoms because the organ was not developed in the right way. So this disease do not obey to a single mutation. They are acquired disease during development. And this is because the microglia remain active. And nitric oxide will be one of the mediators, but there are many others.
Very nice presentation, thank you. I have a quick question. You're looking at Parkinson's, ALS. Have you considered looking at other conditions that may be easier to access? For example, Crohn's disease. We know that, for example, in the gastrointestinal tract, connexins and pannexins play a big role in homeostasis.
Yes, yes.
Perhaps it would be easier to get the compound, the D4 compound, to treat some of these things. And as you know, there's no cure right now for Crohn's. Have you thought about looking at other tissues, and what would be your expected outcome if you were to treat it there?
Yes, I have thought about it, but not done yet the studies. It's difficult to do all the studies at the same time and to have the models, animal models. And one of the problems is to progress with one molecule and to get to human. We need to sell the wonder molecules is not something that the industries are interested in. You need to go one by one with one business. So we have to go and study the toxicity of one molecule first for one treatment. If you say this molecule is good to treat this and this and this other, then the pharmacy is not interested because then you burn the business. So we are making progress with muscle atrophy and with the epilepsy, but we have several other models, and eventually we can, the dream is to study different diseases.
Yeah, normally we think of connexins as a gap junction, no?
Yes.
That makes communication between two cells. What is the relative importance of these hemichannels, and would these be signaling on the apical region of the plasma membrane or wherever, or the basal region? Any idea?
So far,
I didn't know that hemichannels did anything, so that's my level.
right, yeah. So far, it seems that there are two pools of hemichannels that form the gap junctions, and hemichannels that remain in the cell surface in the apical region, and maybe in the basal region, so, but not in the gap junction region. And these hemichannels is a limited amount that opens very infrequently in the physiological condition, but under disease, they open very frequently, and the amount of hemichannels in both, in the apical and the basal region, increase. So, and the other thing is that we have to consider that inflammatory response is a progressive response. So all this is changing time while time is going.